This experiment uses whole-cell MALDI-TOF Mass Spectrometry to accurately analyze the multiple activation states of macrophages. First, derive the macrophages from CD14-positive monocytes, and then stimulate the cells with agonists like cytokines and bacteria. Analyze the samples by MALDI-TOF on a steel target, separating the proteins and obtaining a spectrum for each sample. Results of specific fingerprints can discriminate between various activation states of a single eukaryotic cell, the macrophage, based on a bioinformatic analysis of the spectra obtained by the whole-cell MALDI-TOF Mass Spectrometry.
The main advantage of this technique over existing methods like flow cytometry is that it's fast, easy, and not expensive.
In this experiment, we show that reproducible spectra were obtained from macrophages and that various activation states were discriminated by whole-cell MALDI-TOF. We also obtained spectra from PBMC, which might be useful for the diagnosis of the host response to infection. This method can provide insights into activation states of immune cells. It could also be applied to other systems, such as cancer profiling, to replace, for example, extemporaneous analysis during surgery.
Isolate the cells from leucopaks. In 50 ml tubes, add 15 ml Ficol. Dilute the blood 10-fold in saline. Then separate 30 ml of the diluted blood on the Ficol gradient.